Welcome to another video tutorial on microscopy. Okay, let's get started. Here in this video tutorial mainly we'll study about microscope, its parts and the different types of microscopes, and we'll study in detail about compound microscope and what the parts of it. Okay, yes, a microscope is a device so that magnifies small objects. So yes, as we all know that microscope is a device so which is used to enlarge or magnify the small objects, and also we can see the objects or organisms which we cannot see through our naked eyes with the help of microscope. Okay, so as you can see in the picture over here, so this is called as magnification or enlargement. As you can see here, so this is the image of a root tip which is visualized at 50x. Okay, the same thing when you double up, this is how it looks. So like that, when you magnify at 200x, this is how it looks, 400x, 630x, and at 1000x you can see the cells very clearly. Okay, so and further if you magnify, yes, you can see the components of the cell also very clearly. So this is what is called as magnification. Okay, so this is how the enlargement happens and magnification happens in the microscope. And there is another example over here. As you can see, this is a image of a human blood cell. So in 50x I think you cannot see the shape of the cell, you can just see that there is something, right? So when you double it up, that is 100x, you can see a little better. This is the 200x, 400x, 630x. Again in 1000x you can see the shape and the structure of the cell very clearly. Okay, so this is the difference. So this is what is called as magnification. Okay, yes, the first microscope was developed at about 1590 and was only as strong as magnifying glass. Yes, so I think all of you would have seen magnifying glass. This is how the magnifying glass looks. So the first microscope, no, so it was developed at around 1590. Okay, and so that had capacity as strong as magnifying glass. So now how much can a magnifying glass magnify? It can only magnify at about 2x to 6x. Okay, so the first microscope only had the capacity of a magnifying glass, and then later most complicated microscopes have been introduced. Okay, now I was coming to a little bit of history of microscope. The microscopes used by Antony van Leeuwenhoek during the 17th century was a simple microscope and had only one lens and is equivalent to a magnifying glass. Yes, so I think we all know about Antony van Leeuwenhoek. He's called as father of microbiology, and he is the person who invented microscope, and so he is the one who told that there is a life beyond our naked eyes. Okay, and he's the one who told that uh, there are small parasites, that is animalcules, which you can see through microscope, and he did a lot of investigation to the field of microbiology. Okay, so yes, so he was present in 17th century, and he was the one who designed microscope. In his microscope, no, the first microscope which he developed, he had only one lens. As you can see in the picture over here, this is the microscope developed by Antony van Leeuwenhoek. So this is the lens over here. So it had only one lens, and it was equivalent to magnifying glass. The magnifying power was equal to a magnifying glass. That's it. Okay. And then the early compound microscope were of poor quality and could not be used to see bacteria. Around 1830, a better microscope was developed by Joseph Jackson Lister, father of Joseph Lister. Yes, at around 1830. Okay, so a better microscopes have been developed. So by a person who's called as Joseph Jackson Lister, who is the father of Joseph Lister. Okay, and then modification and improvement of Lister's microscope resulted in the development of modern compound microscopes. Microbiologists use a variety of light microscopes for their work. Yes, so like this, So day by day, so there was improvement in the microscope. Instead of using one lens, many lenses were being used, and the quality of the lens also improved as a result. So today we have a modern compound microscope. And I think yes, microbiologists today use a variety of light microscopes. Okay, so there are different types of microscopes. So as we can see here, so under light microscope, that is the microscope which uses light, no, so there is a bright field or compound microscope, dark field microscope, phase contrast microscope, fluorescent microscope, and so electron microscope is again a different type of microscope. So under that again we have transmission electron microscope, scanning electron microscope. So now we'll study each of them in detail now. So particularly in this video, so we'll focus on compound microscope or bright field microscope. Okay. Yes, then. Okay, compound microscope. So compound microscope are built using a compound lens system where the primary magnification is provided by the objective lens, which is then compounded multiplied by ocular lens or eyepiece. So yes, so compound microscope is a simplest form of microscope which we use it in laboratories. Okay, so I think all microbiological laboratories will have this simple instrument which is called as compound microscope. Okay, so now here it is made up of compound lenses. So what do you mean by compound lenses here? So it's more than one lens. Okay, so usually it will be made up of two lenses over here. So for primary magnification we use objective lens, and apart from that we also use ocular lens which is called as eyepiece. So as you can see in the picture over here, this is a picture of a compound microscope, and so this is a stage, so where the object which has to be visualized will be placed, and this part, no, this is the objective lens. There will be objective lens over here, and apart from objective lens, so there is another lens over here which is called as ocular lens or eyepiece. Okay, so this is a two lens which we usually find it in compound microscope, hence we call it as compound lens. Then the objective lens is the lower lens nearest to the specimen which enlarges the specimen and is also referred to as a primary lens. While yes, so this objective lens, no, so this is the one which is lower. Okay, as you can see this is below, and so this is called as primary lens. Okay, why? Because so it is nearest to the specimen. Now as I told, this is a stage and specimen will be kept here, so this is nearest to the specimen. Okay, and it is the one which focuses the specimen, and that's the reason we call it as primary lens. And apart from that there is another lens which is called as eyepiece or ocular lens. The eyepiece is a top ocular lens, lies nearest to the viewer's eye and also known as secondary lens. They offer two-dimensional visual information. Yes. So apart from that we have a secondary lens which is called as ocular lens. Okay. So this ocular lens is also called as eyepiece because it is nearest to the viewer's eye. You completely focus, you completely see through this ocular lens itself. Okay, and that's the reason it's called as that is secondary lens or viewer's eye. Okay, these are the two main lens which is used in compound microscopes. Okay, so next we'll move on to the parts of a compound microscope. So yes, as you can see in the picture over here, this is the compound microscope and these are the different parts of compound microscope. So let's study them one by one. Yeah, the first one is head or body tube. So this is the head or the body tube. So it forms the top section of the microscope which holds the prisms and eyepiece tubes. The presence of a single head which has only one eyepiece is known as monocular head, while the double head bears two eyepieces. So yes, so this is the body tube, and so here you can see the eyepiece. Eyepiece is nothing but a lens. Okay, and here in this microscope there is only eyepiece, one eyepiece, and yes, you call this as monocular head. So whereas in some other microscopes there will be two heads, two eyepieces can be seen. So I'll show you that picture in the next slide. And then next is the arm. It is a strong and curved curvature used to hold the microscope while moving them from one place to another, and also functions to connect the tube with the base of the microscope. So yes, so now as you can see, this curved part which you can see, this curved part is called as arm. Okay, and so this is where we hold the microscope. If you want to move the microscope from one place to another place, one hand. Okay, so we'll usually be holding this arm. Another arm you're supposed to hold it towards the base here. Okay, and then you're supposed to lift the microscope, move from one place to another place. So hence this particular region is called as arm. And yes, it is also a region which connects the base and the body tube. Okay, and then next is base or foot. The term in itself defines to be the bottom component of a microscope that acts as a prop to support the entire weight of the microscope. This facilitate placing it on a table or a bench. So yes, this is the base as you can see here, and yes, this is a component which is required as a support. Okay, this gives the support and this helps the microscope to be placed on a particular bench or table or any other floor. Okay, so then the next is inclination joint. This facilitates tilting the microscope back for more comfortable viewing. It is a region where a pin can be found at the point where the microscope arm attaches to the base of it. Okay, so now this is the inclination joint. As you can see, there is a pin over here, and this pin is the one which is attaching the base, okay, to the arm of the microscope. Okay, and this inclination joint will help in the tilting of microscope for comfortable viewing. Okay, so next is the nose piece. It is also known as the revolving turret, so which houses 3 to 5 objective lenses and is located below the head of the microscope. It can rotate in either direction to lock the objective lens over the stage aperture. Okay, now so this is the objective lens as you can see. So apart from the ocular lens, so this is the second objective lenses. And so here objective lenses can be revolved, they can be rotated easily, and you can select the objective which is required. Okay, usually in the compound microscope, in the normal simple compound microscope, we can find three objectives, that is 10x, 40x and 100x. So according to the magnification which you require, you can adjust, you can rotate and you can adjust the objective, and then you can focus it onto the slide. Okay, and then next is a stage. It is a platform where sample specimens are usually mounted onto a glass slide for observation. So this comes in a variety of shapes including rectangular, square or circular design. The most compound microscopes include an adjustable mechanical stage height. So yes, again this is the stage. So this is the place where the specimen or the slide which you need to visualize is being placed. Okay, usually the specimen, so which you want to visualize, now you will mount it onto the slide, and the slide is placed here on the stage. Okay, and this stage can be of different shapes. It can be square shaped, it can be rectangular shaped, or it can be circular shaped also. Okay, and most of the microscopes, no, so you can also adjust this mechanical stage height of the stage also can be adjusted. You can bring it up and down. And then next is stage clips. So yes, these are the stage clips here, so as you can find there and here. So after placing this slide, you can clip the slide so that the slide does not move, and so you can observe the specimen properly. And then next is mechanical stage knob. These knobs are two in number, where the former moves the slide from side to side while the latter helps in back and forth movement. Yes, so this is the mechanical stage controls, and they control the slide movement. Okay, sometimes yes, the specimen will be present in between, sometimes it will be present towards the end. So if you want to adjust it and focus, no, so then you need to move the slide. Okay, so this mechanical stage control is the one which helps the slide to move uh, sideways like this, or front and back. Okay, and then next is iris diaphragm. It lies immediately underneath the stage, which controls the contrast by adjusting the amount of light that enters the condenser. Yes, so next immediately below the stage there is iris diaphragm. Okay, so they control the amount of light that enters the condenser. Mainly they control the light, how much light has to go into the condenser will be adjusted by this iris diaphragm. Okay, and then next is condenser. It is situated beneath the iris diaphragm, and so it functions to converge the light coming from the light source and focus it onto the specimen. Yes, so just below again iris diaphragm there is condenser, as the name tells here, condenser. So it is the one which converges, okay, so which brings all the light rays at one point. It converges the light coming from the light force source, and then it focuses it onto the specimen. Okay, and then next is there are two adjustment knobs, as you can see here. This is a chorus adjustment knob, and this is a fine adjustment knob. Chorus adjustment knob, a large knob with a smaller knob in the middle, can be found on the side of the microscope close to the base. This large knob rises or lowers the stage and is used to concentrate an object closely. The chorus adjustment knob should only be utilized with low power or scanning objective lenses. Whereas fine adjustment, these are the small knobs on each side of the microscope which is used to focus on the object precisely. Only the fine adjustment knob should be used for all focusing when using the high power objective 40x lens. Okay, now this coarse adjustment knob is mainly used for focusing. Okay, so it can actually move the stage. Okay, it can lower the stage or it can bring the stage above, and then it adjusts it such a way that you can focus it properly. Okay, and then next is fine adjustment knob. So this fine adjustment knob is for obtaining the precise image. Okay, for obtaining the clarity of the image we use this fine adjustment knob. Okay, and this coarse adjustment knob, no, it is utilized when low power or scanning objective lenses are used. Okay, so whereas in this fine adjustment knob, they are mainly used with high power objectives. Fine. And then next is eyepiece or ocular lens. Yes, this is the eyepiece or ocular lens. It is a lens housed at the top of the body tube through which one can observe the magnified image of the specimen on the slide. The ocular lens has a 10x magnification power. Yes. Apart from these objective lenses, there is a second set of lens which is called as eyepiece or ocular lens. So this is called as eyepiece because through this eyepiece or ocular lens only we will be able to look into the microscope. Okay, usually it will have 10x magnification power, and mainly through this we can focus on to the slide. Okay, now let us know something about objective lenses. As I told, so there may be 3 to 4 objective lens depending on the microscope. Now see, objective lenses, commonly 3 to 4 objective lenses with magnification power of 4x, 10x, 40x and 100x are available in compound microscopes. So that means here 4x, 10x, 40x and 100x is the capacity of magnification, how much it can magnify. Okay, so now yes, 4x is scanning objective lens. The smallest magnification offered by any objective lens is that of scanning objective lens. A typical magnification of scanning objectives is 4x. These roughly provide viewers with enough magnification for a good overview of slide. Yes, the first and the smallest magnification enlargement is the 4x. And then next is 10x. 10x is it can enlarge a little more. The magnification power is more, and we call this as low power objective lenses. One of the best lenses for viewing and examining glass slide samples in the low power objective lens, since it has a higher magnification power than the scanning objective lens. Yes, so this is the best magnification power for viewing and examining the glass slide. Okay, and then next is, if you want the sample to be focused a little more, if you want to learn in detail about it, no, so then you can use high power objective lens, which is 40x. Okay, 4 times greater than this 10x. So this will be the longest objective lens if the microscope has the 3 objectives only. It is employed to see an object in much more detail. The 40x high power objective lens magnifies object 40 times. Yes, and this is the longest objective. If you see the compound microscope, there will be 3 objectives given. So among that, the length of this objective will be a little longer, and so yes, this focuses 4 times, that is 40 times than the original image. Okay, and then next is oil immersion objective lens, which is 100x. These are present in microscopes with 4 objectives which have 100x magnifying power. This requires the addition of a drop of oil immersion on the sample. Yes, so there is oil available, so you need to place the oil on the sample, and then you need to use this 100x objective. Okay, and then illuminator. So illuminator is the light source here. So the condenser is illuminated by an integrated halogen lamp, usually located at the base, and features a dial for adjusting the brightness on one side. However, earlier microscopes featured the mirror, which acted as an illuminator by reflecting the natural light into the base of the microscope. Yes, so there will be illuminator also, which is nothing but halogen lamp. So, and most of the normal compound microscope will also have a natural mirror. Okay, and the natural light would enter into this mirror and it would get reflected. Okay, that is a natural source of life. And now most of the microscopes also will have halogen lamp as an illuminator. Okay, so next is a principle of bright field microscope. So yes, there are various types of microscope as we saw in the beginning. So but here in this video lecture mainly we'll focus on bright field microscope, and we'll study the principle of it. Now as you can see here, so this is a picture. So how the light rays passes, how it get condensed, and how it reaches the eye. Okay, so yes, the light path of a bright field microscope is extremely simple. No additional components are required beyond the normal light microscope setup. The light path therefore consists of the trans illumination light source, commonly a halogen lamp in the microscope stand. Yes, so this is the source of light. Usually it will be halogen light. This is the light source. So now from the light source, no, the light is getting produced. Okay, and this light is now reaching the condenser. Okay, see, which focuses light from the light source on the sample. Yes, so this condenser, as I told, so this condenses the light rays and then it focuses it onto the sample. So it condenses, that means it converges, it captures all the light rays and then it produces onto the specimen. Okay, and then from the specimen the objective lens which collects the light from the sample and magnifies the image. So yes, from here again the light will be magnified onto the objective lens, and then finally it will be magnified onto the ocular lens, which you can see through your eyes, or sometimes in some microscopes yes, camera will be connected, the screen would be connected, and so one can view the image onto the screen. So it is just simple. Okay, so light rays will be produced by the source, that is halogen vapor lamp, so that will be condensed by the condenser and then it will be produced onto the specimen, and this light rays over here will be captured by the objective lens, which will further move on to the ocular lens and which can be viewed by the eye. So this is the principle of bright field microscope. Okay, so next is the operating procedure of compound microscope. So the following points are the steps for operating microscope. Okay, so now how you need to operate the microscope. The lowest power objective lens should be snapped into the place by rotating the revolving nose piece. Place the microscope slide on the stage and secure it using the stage clips. Turn the focus knob to raise the stage as you look at the objective lens and stage from the side. Without letting the objective hit the cover slip, raise it as high as it will go. Adjust the focus knobs to focus the image while looking through the eyepiece. Adjust the light's brightness and condenser's position for the higher amount of light. Okay, it's very simple. Now what you need to do is you will first of all prepare a slide. So you can just take the sample, mount it onto the slide, put the cover slip, and you should place it on the stage over here as it is shown. Okay, so once it is placed on the stage, so you will clip it up so that the slide should not move. Okay, so because there will be other adjustments also and slide might fall off, so that's the reason you will clip it up on both the sides so that the slide shouldn't move, and then you will usually preferably keep the lowest objective first. Okay, now for example, uh, as soon as you bring the sample and when you try to visualize, no, it's better that we start from the lower objective. Okay, so that's why you will rotate this revolving nose piece first and then you will fix it to the lowest objective. If it is 4x, so rotate and place it to 4x, and if it is 10x, no, you will rotate and place it to 10x, and then you will place the slide, and then you will clip it up. So once you clip it up, now you can adjust the knobs over here. As I told, so there is a lot of adjustments to be done, like for example, so you can adjust the stage here if you want it to bring above, or if you want to bring it below, so you can adjust the stage over here. Okay, and then you can also adjust the slide, so based on the sample where you have placed, no, so you can adjust the slide also. You can take it back, you can take it front, you can move it sideways, you can adjust the slide, you can adjust the stage, and also you can adjust the image. Okay, so as I told, there is two adjustment knobs, like coarse adjustment knobs and fine adjustment knobs. So you can use coarse adjustment knob to adjust the mechanical stage, bring it into focus, and then fine adjustment knob will be for the clarity. Once you know the sample is over there, once you're focused with the sample, and now you can bring the clarity of the image by the fine adjustment knobs. Okay, so like this, a lot of adjustments can be done. Then you can also adjust the brightness of the light, how much light has to be produced. You can adjust the brightness of the light also, and you can adjust the condenser's position also. If the more amount of light is required, so condenser's position also can be adjusted. Like that, it's only like placing the mounted slide onto the stage, clipping up, and then it's all sorts of adjustments to be done. Okay, and then when the sample is in the center of the field of vision, move the microscope slide. For the clearest image, adjust the condenser and light intensity after focusing the sample. Using the focus knob, you can switch to the next objective lens once you obtain a clear image of the sample with the lowest power objective. The sample's focus or the condenser and the light intensity may need to be adjusted. Make sure that the objective lens doesn't touch the slide. After you're done, lower the stage, click the low power lens into place, and then take out the slide. Okay, so yes, it's like, so once all the adjustment is made, so once you have focused the image, once you have produced, if you've got the clarity of the image, so you can change the objective now. Okay, so you can take it to 40x if you want, or if you want more like 100x, you can take it to 100x. You can focus again, you can adjust the coarse and fine adjustment knob, you can bring the clarity, you can observe with the microscope. The only thing you have to know is that objective lens doesn't touch the slide, it should not touch the slide. Okay, you can bring as much higher as possible, but you should note that the objective lens is not touching the slide. Okay, that one thing has to be done. And once everything is done, once you have visualized and everything, you have to lower the stage again. Okay, it's not that you have to grab the slide, so there will be chances that the slide might touch the objective and it might crack, and that's the reason you have to lower the stage. Okay, and then low power lens, you click the low power lens into the place again, and then you need to take out the slide. Okay, this is what has to be followed. Applications of compound microscope. A compound microscope is very helpful in pathology labs while conducting blood analysis for disease diagnosis. Yes, a pathology laboratory is a specialized medical facility where various biological samples like blood, tissue, body fluids will be analyzed, and it will be diagnosed for disease, and it guides for treatment. Okay, so yes, so here we use compound microscope for conducting blood and various disease analysis. Okay, and apart from that, it aids in the visualization and understanding of microbiological realm of bacteria and viruses. Yes, you can study in detail about the bacteria and viruses. And then it's also used to determine if minerals are present in or absent, in addition to the presence of metals. And then apart from that, the usage of microscope in academic experiments is advantageous for students in schools and universities, which facilitate them to see bacteria and viruses that are normally undetectable to the naked eyes. Yes, we also use it in school and college laboratories. So we make students see the bacteria and viruses which we cannot see through our naked eyes. And apart from that, it is also used in forensic laboratories. Okay, so that is where human cells are studied under the microscope here. And apart from that, we also can use it for observing and visualizing the plant cells, okay, and observing and identifying the organisms present within them.